Namaskar. Welcome to our channel by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, we did discuss development of male gametophyte and development of female gametophyte, as well as types of pollination and outbreeding devices. Now the topic is development of embryo. After fertilization, development of embryo is concerned. Here, imagine this is a zygote after fertilization, and this is endosperm mother cell. This is endosperm mother cell after fertilization, and antipodal cell are supposed to get degenerated. Egg cell synergids also supposed to get degenerated. So this is zygote, and this is endosperm mother cell. Now topic is development of embryo. So how embryo is being developed? Zygote undergo mitosis to produce two cell stage. One is large cell and another is small cell. This large cell and small cell again divide into two, having one basal cell and one suspensor mother cell. These divide transversely. So to have better understanding, these produce a embryo proper cell. So this is a basal cell, and this one is embryo cell. Now this embryo cell divide transversely. When these divide and produce two cell, these divide and produce two cell, this one forming one large cell. Again this will continue to be a basal cell, and this one is suspensor cell, suspensor cell, and this cell divide transversely, producing two cell stage called embryo. Now basal cell remain large. This basal cell remain large as it is. Basal cell remain large, and suspensor cell divide and redivide, continue to divide and redivide, and produce a long suspensor to push this developing embryo deep into. Now this embryo proper cell also divide and redivide to produce a large globular embryo. So here this is a globular embryo. So here globular embryo. Now again basal cell as a large cells to push that embryo deep into. Suspensor cell also continue to be as it is, with a very cell in contact with this called hypophyseal cell, and now this developing embryo become heart shape. Here you can understand this. So heart shaped embryo, This is the next step. So again this is continue to undergo development, where basal cell remain basal cell. To have the understanding, next to this diagram, basal cell remain large, suspensor cell supposed to get contract by the time begin to contract, and embryo proper, that heart shaped embryo, start to take shape of developed embryo with these two cotyledon and radicle at the base. So here this cell in contact with embryo called hypophysis that form the root cap. This is radicle, this is hypocotyle. Now this part is called epicotyle, and here this is plumule, and these two are developed cotyledon in non-endospermic seed, dicot seed. So here basal cell get degenerated later, suspensor contract, condense and get degenerated, only embryo remain with the seed. So next diagram to have the understanding in the seed is like this. So this is a embryo axis. So finally this matured embryo take the part or take the place in corner of the seed with this endosperm, or in non-endospermic seed these are formed the cotyledon. So this is a seed with this developed embryo, So we did understand here how this embryo is being developed, that zygote towards the end of the micropyle. Here when finally seed developed, that seed has one knock called hilum. That hilum become micropylar end, where it is very close to have diffusion of oxygen and water. So this is a place to open. This was the micropyle, that to have the diffusion of oxygen and water during cell germination. So this knob is called hilum. Yes, this is a developed embryo with developed seed. Zygote undergo mitosis to produce under unequal division to produce one large cell and small cell. Large cell is called basal cell, small is called embryo proper, and this large cell and this one again undergo division. Large cell again form one basal cell and suspensor cell. This embryo proper divide transversely to form the two cell stage. Now basal cell continue to be as it is basal cell, but suspensor cell divide and redivide to form long suspensor to push this developing embryo deep into the endosperm and thereby this long suspensor. At the same time, this two cell embryo proper divide and redivide to form first globular embryo. Now this continue to develop and form the heart-shaped embryo. This continue to develop and form this embryo axis with two cotyledon. Embryo axis containing radical, epicotyle and plumule with these two cotyledon deep into the nucellus for the development by this endosperm. But once this embryo and embryo axis is developed, it take the place at the corner of the seed, and this suspensor later get degenerated and basal cell also degenerated. Hypophyseal cell later, it can be hypophyseal cell that help to develop root cap calyptra over the root tip, which is developed by this radical. So hypophyseal cell continue to be with embryo axis and embryo inside the seed. Thus this embryo is being developed. So it is very easy as a part of concept. I hope you did understand where the embryo is being developed, what is the base to develop the embryo, that is zygote which produce after fertilization, what are the parts of the embryo and how this embryo is being developed in process. Question can be asked, draw a label diagram to show different stages of development of embryo. Even in practical, you are asked to separate out heart-shaped embryo, globular embryo or developed embryo from a given spot or a seed, a raw seed, green seed. So you would be aware about this one. Question can be asked, explain the structure of embryo in heart-shaped structure, or all the steps involved in development of embryo till heart-shaped structure. So be ready with the question for diagram as well as 3-mark question, explain process of development of embryo in flowering plant. If any question you can ask in comment section. I appeal you to subscribe and like our channel as well as press bell icon to receive new videos as and when they are uploaded. Thank you.